Hello, good afternoon. So now we have the second plenary session. We will have a lecture from Dr. Azza El-Remessy about mutations of B75 NTR and the mesenchymal stem cells for the treatment of diabetic retinopathy. Then we have a lecture from Dr. Olina Borshiko about dynamic of susceptibility to antibiotics of microorganisms isolated from patients being on mechanical ventilation in the intensive care unit. And finally, we will have a lecture from Dr. Sameh Al-Saidi about functional porous materials for gas separations and environmental applications. So we will start by lecture of Dr. Azza El-Remessy. Please.
Good evening, everyone. My name is Azza El-Remessy, and I will be the speaker for this session. I'd like first to start by thanking the organizers, especially Dr. Ashraf Khalil and Dr. Sameh Said, for the kind invitation and for making me part of the 7th Congress for the Euro-Mediterranean Conference on Life Sciences, Pharmacy, and Biomedicine. I will go ahead and move to the topic. So let me share my screen with you. So my talk today will be entitled Modulation of P75 Neurotrophin Receptor and Mesenchymal Cells for the Treatment of Diabetic Retinopathy. Are we there yet? So this has been a journey over 20 years of our research for our group, attempting to answer this question. And I will try to give you the very quick run. I promise I'm not going to keep you here for 20 years. I would like to mention that this work was, was done over so many years during my tenure at the UGA College of Pharmacy, the Augusta Biomedical Research Corporation, while my visiting year in the College of Medicine in University of Illinois at Chicago, and currently at the Department of Pharmacy in Doctors Hospital of Augusta. So I'd like to acknowledge this great institution that made this happen. Right. As I said, the disease that I'll be telling you about is the diabetic retinopathy, and it is a leading cause of blindness in the working age of U.S. adults, and it's causing it's the most feared complication of diabetes across the board. About 40% of the 40 years or older diabetic patients, they have some level or some symptoms of diabetic retinopathy. And if we look at the current treatment option for diabetic retinopathy right now, you will see that it's limited either for laser photocoagulation or for intravitreal injection using the anti-VEGF therapy. Both of these are invasive in nature. They are only approved for late stage of the disease. 50% of our treated patients, they relapse, so they become refractory for treatment, and we cannot offer them further treatment option. And one of the biggest issue also that reality, we are just furthering the progression in the damage, but we are not really restoring or preventing the damage for other retinal cell types. So with that you can appreciate that there is a pressure on the eye researcher to develop new effective therapeutics for diabetic retinopathy. Something else other than VEGF, which is the vascular endothelial growth factor, many of you already know, and something that can help with preserving other retinal function, and especially the neuronal function. And that's how I become interested in nerve growth factor, or NGF. So I'll give you a little bit history here. So the first lab really that looked into the use of nerve growth factor and how it would be related to preserving the nerves and the vascular capillary in the eye was the group of the renowned scientist, Dr. Michael Brownlee, back in 1995. And they did a brief study where they supplemented NGF for 3 months in diabetic animals, and that preserved their neurons and their capillaries. Soon after that landmark study came another landmark study by Alistair Barber and his group, where it characterized the neuronal death that happened in early diabetes in the human diabetic retina. And that's something that started and then it just continued to happen throughout the disease. With that, that kind of, ignite the interest more on the role of nerve growth factor and neurons. Later in 2005, another group of clinical researchers looked into the NGF levels. In reality, we were kind of expecting that the NGF level would be less in those diabetic patients. But for the paradox, it was found that there is significant increases in NGF level that was documented in serum, tears, and also plasma of these patients with advanced diabetic retinopathy. So if you put that together, we had, like, as I said, a paradox. You have an increase in a growth factor, but you have decrease in the viability of the neurons under diabetic condition. So there is something doesn't add up, right in that. And that really needed some digging and some explanation. And I was privileged to lead studies that resulted in or attempted to understand that paradox in diabetic patients. So back in 2008, that came for our first paper, where we showed that similar to these studies, that diabetes is able to trigger the NGF expression in the diabetic retina. So to the top part here, you can see that when you look at the NGF mRNA level in these diabetic animals, and these were about 5 to 6 weeks of diabetes, which is more or less about 5 years of diabetes in humans. So that's talking about early diabetes. You'll see that increase in expression of NGF. At the same time, we also we also observe it decreases in number of neurons in the diabetic retina. So digging more, into kind of like what could be the reason of why we see increase in NGF mRNA and we do not see that protective effect, we identified that because of the diabetes oxidative stress and formation of peroxynitrite, it's a powerful nitrating agent, that this caused nitration of the TRK-A, which is the survival receptor of NGF. And along with that nitration of the receptor, you see that the phosphorylation, which is a marker for the activation of the receptor, is completely lost here in the diabetic animal. Treatments that decrease the nitration of the receptor was associated also with partial restoration of the receptor activation. So if you look in here, and as I said, you're more than welcome to look at the publication for full details, that in response to the, diabetic injury, the NGF mRNA level increased. However, the function of the survival receptor, the TRK-A, was impaired because of the nitration. So downstream from there, we looked at the BI3 kinase and AKT activation and the neuronal survival. The other phenomena that we observed from that study was that while we see inactivation or inhibition of the survival receptor, the TRK-A, we see also at the same time impairment. We see also increase in the P75. That's the pro-apoptotic receptor for NGF. And you're going to get to appreciate more when we look into the next slide. So our group was the one that really kind of put the new made the new discovery that how diabetes disturbed the homeostasis of NGF and also its receptor. So you can see here that NGF signaled through the TRK-A, the survival, that we just showed you data that this is impaired under diabetic condition, and because of that you get to trigger the P75. That's what lead to the cell death. So you have twofold twofold of reasoning to, leading to the neurodegeneration. The increase in the pro-NGF and P75, the decrease in NGF signaling in these cells. So another interesting factor that the NGF is usually released as a precursor or a pro. So usually under normal condition, pro-NGF is processed into NGF, and that's how you get the survival signaling. And when we dig a little bit, we found interesting fact that diabetes also impair the processing of pro-NGF, which means that for those diabetic patients, they have high level of pro-NGF, they have low level of NGF. We observed that in patients in the vitreous, from patients with about 25 years of diabetes. From the aqueous humor, we verified that in experimental animals. So we came up with that hypothesis that the processing of pro-NGF is impaired due to impairment one of the proteolytic enzyme, the MMP7. And here you go, we checked the MMP7 activation in these patients, and you can see here that there is a significant decrease in the activity of that proteolytic enzyme. These came really in detail in two publications back-to-back in 2011. And as I said, that kind of, explained for the first time that, why although we see increases in NGF and mRNA, why you see less of the effect itself of the survival. And also another fold was why the pro-NGF is accumulated. And as I said, pro-NGF is our bad guy, NGF is our good guy. So, the diabetes impair the survival at two different levels. One level at the processing of the ligand itself, and the other level is at the receptor level. Fast forward in 2015, we were able to assess the levels, that imbalance between pro-NGF and NGF in the ocular samples from our diabetic patients and compare it to the serum. And it was mirrored in the serum. And we were so excited about that because that means that we can detect that imbalance between pro-NGF and NGF in serum of our diabetic patients and assess the severity of their diabetic retinopathy without really invasive sampling from their, from their eyes. Yeah. The next really, this is just an abstract that, a diagram of the many studies that kind of, we've produced over the past 2 decades. So our lab was able to characterize how pro-NGF through activation of the P75, the neurotrophin receptor, led to inflammation, neurodegeneration, barrier dysfunction, occluded capillaries, which is a surrogate marker for ischemia. And you can see that, the detailed citation here. On the other hand, we also kind of studied how that imbalance of having a low NGF was associated with a decrease of the impaired survival and impaired angiogenesis, which is the ability to repair and form new blood vessels in response to injury. So the next item that I would like to talk to you about, today, that's kind of our more recent. So I'd like today to kind of take more time to talk about some of our recent work, is to talk about the mesenchymal stem cells and how this link to the NGF and pro-NGF balance. So mesenchymal stem cells, they are kind of hot topic right now. They are used for lots of several ischemic disease, including retinal disease. The advantage is that they could be mobilized into peripheral blood. We can collect them from peripheral blood, expand them in vitro, and then use them again back either to the same patient or you can donate them to a different patient. The therapeutic potential of mesenchymal stem cells usually is attributed to their ability to differentiate into different cell types. That's one of the pathways. But however, the ability to secrete really rich factors, or what we call the secretome, is their really powerful side. So the secretome, what the mesenchymal stem cells can secrete, they can secrete neurotrophic supporters, they can secrete immunomodulatory factors, they can secrete angiogenesis modulators. And the advantage is that you can take that secretome and injected back to the patient, avoiding the immune reaction, because that's going to be a cell-free therapy. And we're going to talk a little bit more, about that. So the question would be, should we use modulated MCs, or should we use the plain, mesenchymal stem cells? Yeah. And if you look, this is one of the well-written reviews that if you want to know more about the basics of mesenchymal stem cell therapy, please, you can visit that. You will see that also it's interesting that the B75, our neurotrophin receptor, is also one of the stem cell marker. It's known as CD271. This is receptor for all the neurotrophin, one of the essential markers that it was shown to directly inhibit the differentiation of MCs into different cell types. So in other words, if we can manipulate the B75 expression on these MCs, we would be able to allow them to differentiate and to help them differentiate and be homed and help with the vascular protective or neuroprotective action that we're looking for. So I hope I can give you a sort of a background and a good foundation on what we're going to be talking about today, and that would lead us to the goal or the aim of our work that that I'd like to share with you today. So we were hoping really to evaluate the impact of modulating P75 expression by itself or on the mesenchymal stem cells, and to see how that together they can improve the vascular protective and visual function in the ischemic retina. For that, we elected to use ischemia reperfusion model. This is a relatively easy model that many of you, if you're interested, you can establish it at your lab. The idea is to increase the ocular pressure on, locally in the eye of the animal for about 45 minutes to 1 hour. Increasing the ocular pressure using normal saline will cause really a physical pressure in the back of the eye and in the back of the retina, and that's gonna cause the degeneration of the capillaries. After that, we relieve the pressure, you get the blood flow, reperfusion, and that really mimic lots of the endpoints that we see in the diabetic retinopathy and other ischemic retinopathy disease. That model was validated by Dr. Kern and was published earlier in 2007. It is widely used really by eye researcher. The common endpoints that we'll be looking at, that it caused the same inflammation, oxidative stress marker, cause development of acellular capillaries, which, as I said, that's our surrogate marker for ischemia, and visual impairment. So it's an ideal model for screening therapeutics. And as I said, we have done a lot of work in using diabetic models, but for this one, we wanted to do a quick screen for the P75 inhibitor and for using mesenchymal stem cells as a therapy for diabetic retinopathy. Our first piece of results here showing, trying just to show you here that when you do ischemia reperfusion, you trigger the development of acellular capillary, which, as I said, that's a surrogate marker for ischemia and hypoxia in the retina. You can see that there is a big difference here between when you do ischemia reperfusion in the wild type, and you see partial and significant, protection in animals that genetically lack the P75 neurotrophin receptor. Next, as I said, we wanted to use that model to see if we can inject mesenchymal stem cells and see how that, is protective. So these animals went through ischemia reperfusion, and then 2 days after the injury, that's when we injected 100,000 cells of GFP-labeled mesenchymal cells. And then later on, we assess after 7 days their homing, their incorporation, and their vascular protective effect. As you can see in the next slide, that these cells, 3 days post-injection, they were incorporated into into different places in the retina. After 7 days, you can see that they incorporated and they become part of the capillaries of these retina. And the other shape that you would look for is that kind of amoeboid shape. And they have picked up the stain of the isolectin, which means that's a kind of our indication for that they got incorporated into the native vasculature of the retina itself in the ischemic retina side. So we got our first evidence in 3 days, and it stayed incorporated after 7 days. Now we wanted to see what the effect is. As you can see here, that injection of MCs improved the number of acellular capillary in both wild type and also in the knockout. So it has an additive effect. So the deletion of the P75 give us some protection, and then adding the MCs on top of that give us another layer of protection. Next, we wanted to see if we deleted the P75 expression from these mesenchymal cells and then inject them back, would that be even give them superior effect? So we silenced the P75 expression using siRNA on these cells, and then we injected these cells back into our animals, looking to see how the incorporation. You can see here that the scrambled mesenchymal cells were able to incorporate into the ischemic retina. However, the siRNA mesenchymal cells, which means that it lacked the expression of P75, the number and the level of incorporation and homing were much more, which was significantly higher in these ischemic retina. Next, we looked into the number of acellular capillary. That's our marker for ischemia. And you can see that in wild types that did not receive just received the plain mesenchymal cells, you get still a level of acellular capillary. With the one that received the P75 siRNA treated mesenchymal cells, you get way less. It's almost normalized back to normal with in terms of number of acellular capillary. Next, all of this is, interesting, genetic models. However, we'd like to provide, something pharmacologically effective for our diabetic patients. So we tested one of the compounds that we got through our collaboration efforts with Dr. Frank Longo in Stanford University. The LM11A31, I'm just going to be referring to it as an LM compound. This is the first orally bioavailable modulator of P75, and it's highly selective. It can cross the blood-brain barrier, and it completed phase 1 clinical trial for safety and phase 2 for dose selection in patients with moderate Alzheimer's disease. I'd like also to mention that our work is under, patent. It's under provisional patent, and we are working toward the full patentee application. In the next few slides, I'd like to show you some of the positive effects of this LM compound. We shown that modulating the P75 expression, either using the LL compound pharmacologically or using the siRNA genetically, significantly enhance the secretome of the mesenchymal stem cells. We did look into specific factors. So the expression of the SDF1 receptor that is going to be responsible for their homing. The VEGF is secreted also to help with the vasculature survival. NGF is for neuronal survival. And this is just to show the equal loading using the BOUNCE-OS. The next one going to be showing you also the effect of either the LM compound or the siRNA, how this affect when we take the retinal endothelial cells as an end target cells, and we treated them with the secretome coming from these mesenchymal stem cells. And you can see that the expression of the endothelial cells themselves responded really well to these mesenchymal stem cell treated, and by increasing also VEGF expression, AKT, and BCL2, these are markers for survival. So this means that the vasculature is able to secrete their own survival factors better when they are treated with mesenchymal cells that lack the P75 expression. Next, we looked into the angiogenic response. So we've taken, same cells, same secretome, and we detected here angiogenesis endpoints. Here you can see that you're looking at the cell migration. This is called the wound scratch assay. And the blue label here, this is just a dye that you add to your to the plating of the cells. So you can assess, this was like the cells originally at 0 time, and then this is how they look. They grow beyond that blue line after 12 hours. You can see here that using the LLM compound that you get significant increase in the number of migrating cells. We see also similar effects with using the sRNA. Similar effect we also detected on the tube formation assay. This is an assay, one of the standard procedure for angiogenic assay, on the ability of the endothelial cells to form a tube together, because that's what's going to form the initial retina capillaries before it become rounded with smooth muscle cells and form a mature blood vessel. Lastly, I'd like to share with you that we have done, and this is like the first study to report using the visual cue water maze as one of the measures to measure visual acuity in rodents. So if you're familiar with the water maze, basically we train our animals to swim in a colored water. They are looking for a platform where they swim until they get into the platform and then leave the water on that one. So the animals were trained for 3 days. Then after 3 days, they go through the ischemia reperfusion injury. After the ischemia, after 3 days of the ischemia reperfusion, that's when they start receiving the treatment or the saline. And then we tested them again for how long the swimming time every 3 days. So this way we take this as a clue for their visual assessment. I'm excited to show some of these results. Where you can see here, we're looking at the red line on the top, that these are the ischemic reperfusion animal that did not receive any treatment, they just received the vehicle, that you see increase in the swimming time, that indicate that they're losing their vision. That peaked after 6 days of the injury and kind of stayed plateau through the 15 days of the study. On the other hand, the ischemia reperfusion treated animal with the LM compound, they have that same increase at the beginning of the delayed swimming time, which means that their visual acuity was affected. Interestingly, that kind of improved significantly over the course of the 6, 9, 12, and 15 days. If you look on the area under the curve, this way might be easier on you to look, that you will see that ischemia reperfusion significantly increase the cause delay in the swimming time, or as I said, indication of decline in vision. Treatment with the LM compound significantly improved that. With that, I'd like to just summarize here that we think that modulation of P75, either the activity or the expression, can potentiate the effect of mesenchymal stem cells as therapeutic tool. It also helped to harness the vascular reparative efficiency in ischemic retinopathy, disease. We hope to take the LM compound into clinical studies, soon, to be able really to answer the question that are we offering a treatment for diabetic retinopathy? And as I said, I'm hoping really to see this happening in the next, few years. I would like to leave you. I am hosting a special article in the International Journal of Molecular Sciences. Right now, we are doing the second special issue. It's entitled The Therapeutic Modulation of Mesenchymal Stem Cells for Effective Regenerative Medicine. We extended the deadline until November 30th. So feel free to contact me if you would like to submit a review or a research article or a mini review. I'll be happy to assess you with that. With that, I will leave you, and thank you very much for listening.